Hey everybody. For this, I'm going to begin our conversation of amino acid metabolism. Now, in order to have a good understanding of amino acid metabolism, it's definitely a good idea to recall uh, the basic structure of an amino acid. What's the backbone look like, and then how do those amino acids differ from one another? Now, when it comes to amino acid metabolism, a reaction known as a transamination reaction is absolutely critical to understanding how amino acids get metabolized and how they get turned over into something new. So, as a general structure, here is our amino acid. And, you know, this right here is kind of separate from this right here. So, essentially, what that means is our nitrogen, well, that's a little bit new because if you think about what we've talked about this semester, we've talked a lot about carbon metabolism, and we haven't really talked anything about amino or amine metabolism. And so this basically is kind of the sticking point, really. So the amine of any amino acid is really kind of like I said, the sticking point. Now, an amino acid is going to react with something like alpha ketoglutarate, and on the other side of that, we're going to produce an alpha keto acid and glutamate. Now, if we compare the structures of our amino acid and our alpha keto acid, the difference between them is rather than having an amine, we have a carbonyl carbon. And then for alpha ketoglutarate, rather than having a carbonyl carbon or carbonyl oxygen, we have an amine. Okay, so that amine has just been moved from one molecule to the other. Now, this is a reaction that is going to require PLP. PLP is a cofactor. You could kind of think about it like how we've thought about biotin and how we've talked about biotin as a coenzyme that drives this reaction. Now, what I'm going to expect of y'all is that you know the structures that are discussed in lecture as well as in any sort of video supplement that I provide y'all with. Essentially, most of the ones that we're going to talk about are the pretty easy and straightforward ones. We are also going to begin our discussion of a cycle known as the urea cycle. And so anything that pertains to the urea cycle, definitely look at that and say, okay, that's something that I need to know. Because that's going to be something that's not necessarily outside the scope of the transamination reaction, but whenever you think about amino acid metabolism, what I want you to do is think urea cycle, transamination reaction. Those should be kind of your headlines, if you will. And any reactions that require PLP or vitamin B6. You should definitely know those structures and know those reactions. Now, why do we actually basically metabolize amino acids? And where are they coming from? Well, first and foremost, they're coming from proteins. So, why do we have protein turnover? So, why do we degrade a protein? One reason that's shown here is in order to remove abnormal proteins. So proteins that maybe have misfolded and haven't been refolded, proteins that aren't really carrying out the reaction that they're expected to carry out. Another reason for protein turnover is to basically utilize and free up any sort of nitrogen that's been stored. Then, one other factor is regulation. So, regulatory proteins often have short half lives, so that they have a timeline that's not needed kind of all the time. One example that I think of with respect to like degrading a protein for the purposes of regulating it is I always think about our proteases. Now, this is kind of tricky because proteases are the enzymes that break up proteins. But I'm thinking about how long do you want a protease to be around in a cell? Now, if you remember, proteases are activated by first their zymogens. Those zymogens undergo proteolytic activation or proteolytic cleavage, which is just going to lead to an active protease. Now, what I'd like to do is focus here on these zymogens. These zymogens are they're free, they're in a cell, but they're not doing anything. They're not able to, I mean, they are inactive proteases, so they can't actually break any peptide bonds. So they're kind of sitting in wait, and whenever the time comes, then a protease will come along and check them, cut a peptide bond, and activate them. So essentially, what that means is that this protein is there, it's not doing anything, but then it's activated. Now, just as it's activated, you also kind of want a mechanism to turn it off. And it's not going to be turned off by phosphorylation or anything like that, some other mechanism, but instead, you're going to have to basically break up a protease. So you're going to have to consume, chew up a protease. So regulatory proteins like proteases, as an example, are going to have short half-lives. So they're going to get degraded pretty quickly. Now, one way in which a protein can be degraded is through a process known as lysosomal degradation. Now, lysosomal degradation, as you might guess, involves a membrane-bound organelle known as the lysosome. So essentially, proteins get degraded in the lysosome. Now, there's kind of a bit to that. First and foremost, this involves proteases. So this involves proteins that are going to break down peptide bonds, that are going to break peptide bonds. Now, these proteases, they have an optimal pH, so they are most active at about 5. So at a pH of 5, this is a very acidic environment. Now, environment. With that in mind, when you think about the reason for a membrane-bound organelle is the environment inside a membrane-bound organelle is going to be different from that outside of that membrane-bound organelle. And the lysosome is a perfect example to illustrate that, because to align with these proteins being most active at a pH of 5, the lysosomal pH is about 5 to 5.2. So a very acidic environment, which aligns with this aligns with these proteases that are most active in that environment. Now, with respect to lysosomal degradation, we have this process is non-selective in well-nourished cells. Now, under starvation, so in limited resource conditions, we have selective degradation of certain proteins. And these certain proteins have this polypeptide sequence. This polypeptide sequence is KFERQ, or lysine, phenylalanine, glutamic acid, arginine, and glutamine. Now, this is selective degradation of KFERQ proteins in the liver and the kidney, but not in the brain. So these proteins, you could kind of think about it as, or the way that I think about this is, in a cell that is starved, a liver cell or a kidney cell that's deprived of nutrients, well, there's going to be a process of like, okay, well, we've got to maintain this level of blood sugar. We've got to maintain some like available resources so that this cell can continue to exist. What are our options? Basically all hands on deck almost. Well, we're going to kind of have a triage, like what are the proteins that we can get rid of and still kind of function, still exist. And what ultimately happens is these proteins with this polypeptide sequence are targeted. And the way that I always kind of rationalize it is that if a polypeptide has this sequence, KFERQ, that means that protein is basically not essential for cell survival. It's kind of like, it's equivalent to, I don't know, the hair or something like that. I don't need my hair if I need to survive, I can get rid of my hair. If I need to survive, I can get rid of proteins that have that specific pentapeptide. Now, let me be clear, or one thing that I'd like to add is that this is something that, 25 years ago was a bit more like, yeah, absolutely, every single KFERQ protein is going to be degraded. But things have changed a little bit, and more evidence supports that it's not really an absolute, not 100% of proteins with KFERQ are going to be degraded, but it still is a disproportionate or a higher percentage of proteins with KFERQ are going to be degraded. Now, lysosomal activity is going to vary a little bit. So that lysosome is going to be more active in muscle wasting. So in a disease state or an injury or a lack of use, so muscle atrophy, you're going to activate lysosomal degradation. Now, there's also certain physiological circumstances. Regression of the uterus is going to initiate this lysosomal degradation. It's also increased in arthritis and certain, I believe it's in certain autoimmune effects or autoimmune diseases. Now, another type of proteolytic degradation, I would say that, lysosomal is number one, this would be number two, is our proteasome. Now, proteasomal degradation is a little bit different from lysosomal degradation. For starters, proteasomal degradation is driven by this protein complex right here. This thing that is made up of a 19S cap, another 19S cap, and this 20S kind of central unit. This whole thing combines to make something known as the 26S proteasome. The 26S proteasome, this protein complex right here, well, it's pretty interesting because unlike lysosomal degradation of proteins, in order for a protein to be degraded by the 26S proteasome, here's my protein, it has to receive a tag. In that red circle that I've drawn there, that's my tag. And then once it gets tagged, then it will be basically migrated towards this cytosolic protein, and it will be cleaved up into short polypeptides, and then these individual tags right here will be released. Now, I want to talk about a couple of things. For starters, this 19S and 19S and 20S, that has to do with a process or a technique known as analytical ultracentrifugation. Now, centrifugation you did in your Gen Chem 2 lab where you spin things really fast, and I think that there are some folks who probably have some familiarity with centrifugation from the perspective of, what's it called? Clinical centrifuges, separating the components of blood, for example. Well, analytical ultracentrifugation, this is a technique that spins something super fast. We're talking about 80,000 revolutions per minute. I was going to say second, but that doesn't make any sense. And it's going to go at an extremely high rate. And you're going to separate things just as you do with clinical centrifuges. But you're going to separate based on a couple of different properties like density. And then in addition to that, you're using different solvents to get things to separate differently. Now, with that in mind, what these different components do, the 19S and the 20S. The 19S caps, what they do is they select and unwind ubiquitinated, or I'm just going to write this term tagged, proteins for degradation. Now the 20S, this chunk in the middle, what that's going to do, that's going to take these tagged proteins that have been unwound by the 19S caps, and it's going to break them up into 8 amino acid long fragments, and the tags, the ubiquitin tags are going to be released. So essentially what this is we're going to break up these proteins into short fragments. So if you have a protein that's 244 amino acids, well, it's going to be broken up into lots of 8 amino acid long chunks and UBQ, or ubiquitin. Now, those 8 amino acid long chunks, those are going to be something that's a little bit easier to process by the remainder of the cell. But the 26S proteasome is breaking them up into those 8 amino acid long fragments. Now, let's talk about the tag, ubiquitin. Ubiquitin is the tag that we use to basically apply to a protein that we want to degrade. Ubiquitin marks a protein for proteasomal degradation. Ubiquitin itself is a 76 amino acid long protein. Now, this protein being 76 amino acids, think about that term, protein versus polypeptide. If something is 50 amino acids, it's still largely considered a polypeptide, but once you exceed 50 amino acids, then you're talking about a protein. So ubiquitin is 76 amino acids. Now, that is what we add to a protein for degradation to kind of signal like we need to degrade this. A great question then is which proteins actually get ubiquitinated? Is this every protein? Is this only some proteins? I mean, we saw with lysosomal degradation, we have the KFERQ peptide. Well, when it comes to ubiquitin, we look at the polypeptide sequence. We look at the sequence of that protein. And one of the things that we observe is that there are some hallmarks. There are some sequences that do tend to get a protein tag more often than not. One is this PEST sequence. That's proline, glutamic acid, serine, and threonine. So if a protein has a PEST sequence, chances are it's going to get tagged and ubiquitinated and potentially degraded. Now, those PEST sequences are indicators that it's going to be degraded a little bit more rapidly than others. So think about those as like triaged as like, let's move these along, let's get rid of these. Now, there are other proteins that get ubiquitinated, and they maybe don't follow that much of a pattern. Now, one thing that I do want to say, though, is that ubiquitin is added to a lysine residue. So the single letter amino acid code lysine for K, or K for lysine. That is where we're going to add our ubiquitin. Okay, so if a protein does not have a lysine, it can't get ubiquitinated. So it won't get ubiquitinated. Now, there we go. How are we actually going to do this? I want you to know the enzymes that are involved in this. First and foremost, the enzymes that are involved in ubiquitination of a protein, they are of the class of enzymes known as ligases. So they make bonds between, or they make covalent bonds. And generally they're going to use ATP to make those bonds, but they're not a transferase, so they're not adding a phosphate to a protein. Now, this is the whole rundown. These enzymes are E1, E2, and E3. The names are E1, ubiquitin ligase, E2, ubiquitin ligase, and an E3 ubiquitin ligase. So let's go ahead for just a moment. Let us look at just this top portion. So what I've kind of selected is we have ubiquitin and a carboxy group. That carboxy group is found on the C termini of our protein, of our ubiquitin. And then what we've also got is a sulfhydryl group on our E1. So E1 with a sulfhydryl group is going to then bind to ubiquitin and ATP. When it binds to ATP, it's going to break one of the phosphoanhydride bonds to release inorganic pyrophosphate, the PPI, so beta and gamma phosphates. And what's going to be left behind is AMP. So we're breaking a high energy phosphoanhydride bond. And the reason for that is we're going to harness that energy to make this bond right here between our E1 and our ubiquitin. Now, with that in mind, what this means is that we have made a covalent bond between ubiquitin and E1. So basically E1 says, I'm going to add this to something else, but first I need to add it to myself. And I'm going to hydrolyze ATP in order to do that. Okay, so that's phase one, where E1 is now covalently bound to the ubiquitin. E2, again with a sulfhydryl group. So we're looking at probably what amino acid there? If it has a sulfhydryl, it's probably going to be a cysteine residue. So that E2 is going to come along and find the ubiquitinated E1, and it's going to transfer the ubiquitin to itself. So what that's going to mean is rather than, like, E2 is basically going to displace E1. And we're going to, at the end of step 2, have a ubiquitinated E2. Okay? Now, that's going to release E1, so it can go back up here. And the enzyme E1 ubiquitin ligase can start this process over again. But here we are. We've got our E2, which has been ubiquitinated. We have identified our protein that we want to basically target for degradation. The E3 ubiquitin ligase is going to say, hey, ubiquitinated E2, come along here. I need to transfer this ubiquitin from you onto my condemned protein, onto the protein that I want to degrade. This protein that I want to degrade has a lysine residue, and what I'm going to do is I'm going to make an isopeptide bond between my C termini of ubiquitin and this lysine residue. Now, what I want to make clear here are a couple of things. Of all of the ubiquitin ligases involved in this, E1 and E2 at some point in time have a ubiquitin, or they have been ubiquitinated, but they're more or less carriers of ubiquitin. Our condemned protein is also something that's been ubiquitinated. So you could say E1, E2, and our condemned protein all get ubiquitinated. All get ubiquitinated. Okay, now E3 never gets ubiquitinated because it uses E2 and it transfers the ubiquitin from E2 onto our condemned protein. So that's one of the differences between E1, E2, and E3. E1 and E2 get ubiquitinated, E3 does not. And it's an isopeptide bond that's formed between those two. Now, ubiquitination, it's a very dynamic process. Ubiquitin can be removed by breaking that isopeptide bond. The bond that bond which is broken is done by an enzyme known as an isopeptidase. Now, addition of a single ubiquitin does not trigger degradation. And I think that's an important point to remember because, I started this conversation, I said, oh, ubiquitinated protein is going to get degraded. Well, what about E1 and E2? How do those not get mixed up in the fold and accidentally get degraded? Well, the reason for that is because they only take on one ubiquitin. So E1 and E2 get a single ubiquitin added to them. Okay, so monoubiquitination can be used for other types of regulation. Doomed proteins or condemned proteins usually have multiple ubiquitins added to them in the form of typically a polyubiquitin. Now, I want to go ahead and break this down. First of all, right here, monoubiquitination can be used for other types of regulation. Let's think for a moment of post-translational modifications, PTMs. Now, there's a couple of different PTMs, post-translational modifications, that we've talked about. We've talked about phosphorylation. Okay, and we've talked about methylation. There's ATION, there's acetylation. There's a number of different post-translational modifications where we add a group onto a protein, add a group onto an amino acid on a protein. Now, in the case of phosphorylation, well, if you add a phosphate group to, say, a serine residue, what do we know about a serine residue? It looks a little bit like this, CH2OH. Okay, so there's a serine residue, but if we make it a phosphoserine, oh, it changes everything. It goes from OH to OP, O, should be a single bond there, O. Oh man, look at that. Oh, it's gone from a polar uncharged group to a polar negatively charged group with 2 negative charges. That's going to change the sorts of interactions that this amino acid is going to participate in. Absolutely true. And in some cases, that can take a protein from an inactive fold to an active fold. So it can activate a protein. Okay, so you've replaced one atom with basically 4 other atoms. You've taken something that's uncharged and you made it negatively charged. Man, that has a pretty dramatic effect. Well, now let's think about ubiquitination. A single ubiquitin subunit. A single ubiquitin subunit, 76 amino acids. Now, if we have a protein right here that has a lysine residue, and to that lysine residue, you add a 76 amino acid long protein to it. I always kind of think about this as like, if it's almost like you're walking around, let's say that you're the protein. You're the protein and you are 150 amino acids long, okay? As a protein that is 150 amino acids long, someone comes along and they say, hey, I'm gonna modify you. And they proceed to basically add, I don't know, like 2 adult length limbs, 2 legs to your back. And then they say, okay, well, there we go. Those legs might weigh half as much as your body does, which is equivalent to what's happening here. We're adding this massive thing onto you. What are the consequences of that? Oh man, well, that's going to definitely change how you function in the world. So a protein that's, say, 150 amino acids, if you add 76 amino acids to it, that protein has gone up almost 48% in molecular weight. Is that going to change the function? Totally. In some cases, that protein or that enzyme's activity is going to plummet. That's one factor. Another case, or another possibility, is that it gets relocalized. So even if a protein doesn't get targeted and condemned and ultimately get degraded, that protein's function is absolutely going to be impacted if it gets a single ubiquitin added to it. Now, doomed proteins that are condemned for degradation, they're going to, let's say, here's my protein. Typically, what's going to trigger degradation is something like this. There's one ubiquitin that's been added. To that ubiquitin, another ubiquitin is added. A third, and then a fourth ubiquitin can be added. So this polyubiquitination, so basically a chain of 4 ubiquitins, that is going to be the trigger that basically sends that protein to be degraded. Now, that, I'm going to say that in comparison to something like this. So this has 4 ubiquitins, but they're not in a chain. So it's less likely that that protein is going to be degraded. Now keep in mind, this is dynamic. It's temporary, it's transient. So these 4 ubiquitins could be added and then a chain could be built off of each one of them. And then by adding all of these ubiquitins, that's going to be a good target for degradation. Okay, so when a protein gets ubiquitinated, it goes to the 26S proteasome, it gets broken down into free ubiquitin subunits and 8 amino acid long peptides. Those 8 amino acid long polypeptides are going to be broken down and free amino acids are going to form. those free amino acids are going to undergo a transamination reaction where they'll react with an alpha keto acid like alpha ketoglutarate and produce another alpha keto acid and glutamate. This is going to require PLP. Now, in order for this to happen, it's important for you to remember where on Earth is the 26S proteasome. The 26S proteasome is in the cytosol. These free amino acids are going to make their way into the intermembrane space. And if we take an amino acid like glutamate, glutamate in the intermembrane space is going to be able to undergo a transamination reaction where it's going to be, an amino acid like aspartate can undergo a transamination reaction where it transfers an amine onto a molecule like alpha ketoglutarate, making glutamate. So if we've got aspartate free in the cell, that's going to transfer to make glutamate. That glutamate can go in this direction and make its way into the mitochondrial matrix, indicated up here in the upper right-hand corner. The glutamate can go through this glutamate aspartate transporter, so glutamate will be transported in while aspartate is transported out. That aspartate can then, the aspartate has already undergone that transamination reaction with the help of the enzyme aspartate aminotransferase. The other output from the aspartate aminotransferase is oxaloacetate. That oxaloacetate is going to be able to accept electrons from NADH. NADH is going to get oxidized while oxaloacetate gets reduced, and we're going to generate malate. That malate can go from the intermembrane space into the mitochondrial matrix with the help of the malate alpha ketoglutarate transporter. That malate that is now in the mitochondrial matrix can get oxidized, and then lo and behold, we've made NADH. Now this is the part of this transporter that we've already talked about. We talked about the malate aspartate shuttle, and we largely stayed on the top half of this. So basically from here and above. But now we're changing the way that we're looking at it, because now what we're focused on, or one of the things that we're adding to it, is this amino acid, this glutamate, making its way into the mitochondrial matrix. That glutamate can then itself undergo an a transamination reaction where it's going to transfer the ammonium onto aspartate. So we're going to be able to take the oxaloacetate. Okay, so what has happened is malate has become oxidized. We've generated NADH. When malate is oxidized, we make oxaloacetate. Glutamate and oxaloacetate undergo a transamination reaction. Glutamate transfers a nitrogen, or that NH3, onto oxaloacetate, making aspartate. Aspartate can go out, and glutamate can come in. And you can kind of think about this as, in part, a vehicle to ultimately get NADH into the mitochondrial matrix. But another component is this is a way that we can turn over nitrogens. So keep in mind where things are. The 26S proteasome in the intermembrane space. Within the mitochondrial matrix, that is where we have our glutamate and alpha ketoglutarate and our aspartate aminotransferase. So the mitochondrial matrix is going to take oxaloacetate and glutamate to make alpha ketoglutarate and aspartate, whereas the cytosolic aspartate aminotransferase is going to take aspartate and alpha ketoglutarate to make oxaloacetate and glutamate, so that we can then move NADH into the mitochondrial matrix. Now, transamination reactions occur in two stages. The first stage is when we take an amino acid like alanine and the other reactant of alpha ketoglutarate, and we're going to do our transamination reaction producing pyruvate and glutamate. In order for this to happen, we have alanine and PLP. PLP is our vitamin B6 molecule. We're going to make, in step 1, pyruvate and pyridoxamine phosphate. In stage 2, or phase 2, this pyridoxamine phosphate and alpha-ketoglutarate are going to react to make glutamate and PLP. So that PLP is our input and our output. We have alanine, which is the source of our nitrogen, and glutamate, which is the product of our transamination reaction. So here are some of the molecules that we're interested in. Pyridoxine, or vitamin B6, and pyridoxal 5-phosphate, also known as PLP. Now, the differences between these is that, well, for starters, there's a phosphate group on our PLP, where there is absent a phosphate group on our vitamin B6. I want you to be able to recognize these structures and differentiate between these structures. So your biggest points are the phosphate found here compared to this hydroxide group, then this carbonyl carbon versus this alcohol right here, and then ultimately our double bond placement in response to that. This molecule right here is one of the central parts of this reaction and how it takes place. This is our enzyme, our transaminase enzyme, which forms a Schiff base with PLP. So think about this basically as our PLP. Let me redraw that. Essentially, that's effectively our PLP, and then the top half of this is our enzyme that we form this Schiff base with. Now, that Schiff base must be present in order for our alpha amino acid to react with it. We're skipping over this diamine intermediate, and instead we're going straight over to the amino acid PLP Schiff base, the aldamine. Essentially what we're having take place is this amino acid is displacing our enzyme. Now, by displacing our enzyme, we're going from a Schiff base to a Schiff base. But it's an enzyme PLP Schiff base versus an amino acid PLP Schiff base. So all of the major players, all of the different species and their charges are largely the same, except rather than a lysine residue forming a bond with our PLP, instead it's the amino termini of our amino acid that is our that forms our bond with our PLP molecule. We've got our alpha carbon right here. We've got our alpha carbon right here. Now, what we want to do here ultimately is we're really liking the idea of removing our alpha hydrogen. And it can be removed. And we have this resonance stabilized intermediate. Our enzyme kind of stays by because that's going to help our reaction actually take place. But we have this aldamine resonance stabilized intermediate. Now, that resonance stabilized intermediate is redrawn right here. What's going to happen is we have this ketamine intermediate where we're going to use a hydroxide ion as a base. And what that is going to do is basically rearrange some of the atoms in our ketamine intermediate to form a carbanolamine. So this hydroxide pushes the electrons to our alpha carbon. Our double bonded alpha carbon, this particular intermediate, we're going to move those electrons down so that our nitrogen, which previously was positively charged, is no longer positively charged, giving it a lone pair of electrons. Then we're going to have something, this carbanolamine, that's a little bit more linear. This ketamine and this carbanolamine, what we're going to want to do after that is basically release this amino acid, or what remains of our amino acid. And when we do that, we form an alpha keto acid. So essentially the purpose of this was to get the nitrogen that was on our alpha keto acid and link it to our PLP. And whenever we link it to our PLP, what we're able to do is release this kind of chain of atoms from our PLP. So this right here, release that in the form of an alpha keto acid. So the purpose and the value of this right here is this is going to be able to be our carbonyl oxygen. So that's going to be the donor that acts as our carbonyl oxygen. Now, this is part of our reaction. So this is where I don't know that it's tricky, because if you remember, we had the slide that was stage 1, I think I'll label it as S1, was alanine, which was just given as a general amino acid, plus PLP is going to produce pyruvate plus pyridoxamine, which I'm going to write as PMP. So essentially what we've done is we've arrived right here. This alpha keto acid would be something like pyruvate. So we've gone from here to here. Okay, so that's stage 1. Well, stage 2 is PMP plus alpha KG, alpha ketoglutarate. That is going to reversibly form glutamate plus PLP. Okay, so in the next slide it will make a little bit, or I hope that will make a little bit more sense. Here's everything put together. This is the input on stage 1, and this is the output on stage 1. So as we complete that stage 1, S1 that I said, we've gone from here to here. Now, to get stage 2, we have to go from here back to here. Now, what differs is we're going to basically change or get a different alpha keto acid so that we can make a different amino acid. So think about, with all of these different intermediates, think about this as an intermediate in the sense that it is the end of stage 1. And then we started in the top left-hand corner. We're going to end stage 2 right here. Okay, so our output is a PLP, or an enzyme PLP shift base and an amino acid. Our input is an enzyme PLP shift base and an amino acid. What I want y'all to know, and what you need to know about this, is I need you to be able to recognize a couple of structures. One structure is this right here. Another structure is this ketamine. In addition to knowing what the ketamine looks like, you absolutely need to know what's the purpose of that hydroxide. It's acting as a base, and that is going to give us ultimately that carbonyl oxygen. So I want you to be able to identify the enzyme PLP shift base, ketamine, and the enzyme PMP, and an alpha keto acid and an amino acid. So the things that I'm circling right here, these are the things that I want you to know about, and I want you to be able to identify those structures. And the entire purpose of this was to transfer an amine from one molecule to another molecule, to take an amino acid and make an alpha keto acid, and to take an alpha keto acid and make an amino acid. So that's what we've got with PLP. It is basically our our molecule that allows us to release an amine from an amino acid, and then to put an amine onto an alpha keto acid to make an amino acid. Now, with that in mind, with all of that said, if you look at this figure right here, I'm going to draw kind of a little boundary. We've largely stayed here. We've talked about the synthesis of carbohydrates and sugars. We talked about the degradation of sugars. We talked about the synthesis of lipids in them and the degradation of lipids. Now, what we've just done is we've brought amino acids into the fold. Now, this might seem like a little bit of a stretch, but because we haven't used this term very much, we've talked about these molecules though. Alpha keto acids. We have talked about alpha keto acids because alpha ketoglutarate is an alpha keto acid. Pyruvate is an alpha keto acid. Now, if I asked you how to metabolize pyruvate, would you have a sense of how to do that? Yeah, especially if it's taking place in the cytosol. Well, if you're making pyruvate in the cytosol, what are you going to do? You're going to plug it into right here, and it's going to go through the citric acid cycle, and you're going to be able to break that down into CO2. Okay, so there's that. We've kind of brought in amino acids into that fold. So when it comes to major biological molecules, we have a means to break them down. Now, what's kind of neglected in all this, however, is the ammonium, NH4+. Well, we're going to metabolize that in a cycle known as the urea cycle, and that is going to be excreted ultimately. So an enzyme known as glutamate dehydrogenase removes ammonium, or NH4, in a process known as oxidative deamination. What this is going to do is it's going to use the amino acid glutamate, which, if you remember, that's the amino acid that we were producing. Glutamate plus NADP+. Now, fair question, is this the oxidized or the reduced form? Well, this is our oxidized form. Then, if we look at our products, we're going to end up with a reduced form. So we are going to carry out an oxidation reduction reaction where glutamate gets oxidized, so it's going to give up electrons. NADP+ is going to pick those up and it's going to form the reduced form of it. Glutamate is going to get oxidized and it's going to release an ammonium. So when it gets oxidized, it makes alpha ketoglutarate and ammonium. Now, this is a reversible reaction. And this enzyme, glutamate dehydrogenase, has a high Km for glutamate. So this is a low GLU affinity. Now, our process looks a little bit like this. We are going to, first and foremost, oxidize glutamate, and we're going to make that NADPH, or the NADH. So when you see something like this, that's an indication, with the P in parentheses, that's an indication that this enzyme's a little bit fluid. It could use NAD+, or it could use NADP+. Now, with that said, we're going to make this alpha aminoglutarate. So that alpha aminoglutarate is going to have a double bonded NH2, so a positive charge there, and we're going to be able to lose that. And when we lose that, we're going to make alpha ketoglutarate. So our alpha ketoglutarate is going to result from this. So ammonium, NH4+, is toxic, and one thing that it can do is cause you to become very lethargic. So you need to have a means to get rid of that ammonium. Fish, well, they will just excrete NH4+, done. Mammals produce urea, you need to know that structure. Birds and reptiles produce uric acid crystals. Now, mammals, that's what we're going to focus in on, because what mammals are going to do is they're going to make this urea, and they got to figure out a way to get rid of it. Or they will have a way to get rid of it. Okay, so here we go. This is the urea cycle. One thing that I want you to recognize right off the bat is that this is a colorful slide, a colorful series of pictures. What I want you to recognize first and foremost is that this blue kind of oval is our mitochondria. We already talked about the malate aspartate shuttle and how we were able to move molecules into the mitochondria, move them out of the mitochondria, moving our aspartate and alpha-ketoglutarate in and out. Now, what I want you to do is recognize that this process takes place in the cytosol and in the mitochondria. Now, what we're going to do is we're going to start up at the top. We have an alpha keto acid and an amino acid. This is just a general amino acid. What we are going to do is we're going to carry out our transamination reaction. In that transamination reaction, we're going to produce glutamate. That is going to be used by glutamate dehydrogenase. That glutamate dehydrogenase that generated our NADPH or our NADH. Well, how can we use that? NADPH in the cytosol? Ooh, you know what we can use that for? Fatty acid biosynthesis. Because, well, it's in the cytosol. It's where fatty acid biosynthesis is going to take place. We're also going to generate alpha-ketoglutarate. The most important part, however, is that NH3, or our, I can't zoom in. That NH3 or that NH4, that ammonium or that ammonia, we're going to get that into the mitochondria. And when we get that into the mitochondria, we're going to use an enzyme known as carbamoyl phosphate. What carbamoyl phosphate is going to do is it's going to take two ATP molecules, HCO3 and NH3. And what we're going to do with that is we're going to make this molecule right here, H2N COPO3 2 minus. So if you look at this, it kind of looks like urea. It looks like half of urea, but then we've got a phosphate. It's carbamoyl phosphate is what this molecule is called. We're going to generate two ADP and a single inorganic phosphate. But we had two ATP. Where's our other phosphate? It's right there on our carbamoyl phosphate. So that carbamoyl phosphate is going to then react with a molecule known as ornithine. Ornithine is an amino acid, and what we're going to produce from the reaction between carbamoyl phosphate and ornithine is another compound known as citrulline. Citrulline is shown right here. Citrulline has our carbon right there, and then our nitrogen right there. This effectively is our parts of ornithine that were used to make citrulline. Now this has all taken place in the mitochondria. Citrulline is going to be transported out of the mitochondria into the cytosol. That citrulline, this molecule right here, which I'm going to redraw over here, that is our citrulline. That is going to react with aspartate, and in an ATP-dependent fashion, it's going to produce arginosuccinate. So we have citrulline and aspartate coming together to make arginosuccinate. Now, it's important to keep track of some of the atoms. So if we look here, and you might want to look at this on your own PDF version of this, but here we've got our carbon and our NH2. That NH2 has a positive charge. The atoms that are highlighted in kind of a brownish color, those are all coming from aspartate, and those that are in black are all coming from ornithine originally. This arginosuccinate, this is in the cytosol. Arginosuccinate is going to be metabolized to release fumarate. Fumarate is going to be able to be metabolized into malate, oxaloacetate, and then on back to aspartate. Now, what I want you to know about this is that arginosuccinate is going to release fumarate. What's also going to be left behind is an amino acid known as arginine. That arginine is where we are actually going to hydrolyze away our urea and produce ornithine. So arginine gives way to ornithine, ornithine gives way to citrulline, citrulline to arginosuccinate, to arginine, to ornithine, to citrulline, to arginosuccinate, to arginine, ornithine, citrulline, and so on. Now, this is a fairly complicated pathway. I'm not going to, say that it's anything but that. But I want to go through and break down some of the parts that are most important for y'all to be familiar with. Before we do that, in a nutshell, this is what's happening. We have, starting on the left-hand side here, we have amino acid number 1 that's going to be used to produce an alpha keto acid and glutamate, because that amino acid number 1 is going to transfer its nitrogen, onto alpha ketoglutarate to make glutamate. That glutamate is going to be acted upon by glutamate dehydrogenase. If there's one enzyme that's super important for the urea cycle, it's glutamate dehydrogenase, because that's going to get our nitrogen into the mitochondria. That NH3 is going to react with HCO3-, which is a product of carbonic anhydrase, H2O and CO2. That carbonic anhydrase is going to then, with the help of aspartate, give us our urea. What else is going to be produced along the way? Well, fumarate. Fumarate will then be able to cycle through the citric acid cycle to make oxaloacetate, which can undergo a transamination reaction to produce alpha ketoglutarate, because it undergoes a transamination reaction with glutamate. Then we've got alpha ketoglutarate that undergoes our transamination reaction to generate this glutamate from amino acid number 2, and we're going to get our second alpha keto acid. So that's in a nutshell what's happening. This is something that I would recommend processing as individual components over a series of a couple of days. For instance, the first thing that I would get straight is this chunk right here. I would say that just because that's kind of the easiest part to do, and you've got a natural stopping point if you've got to get that nitrogen, that NH4, into the mitochondria. Now that NH4, that's kind of a part of another block. Well, once in the mitochondria, that NH4 is going to react with HCO3- and aspartate. Then that is kind of my day 2 thing, and then my day 3 is going to be, well, I've got to get that into form my urea. Then the last part is basically the components that lead to the regeneration of that glutamate so that reaction can continue taking place. So this is definitely like kind of an action-packed pathway. Just as glutamate dehydrogenase is an important enzyme, carbamoyl phosphate synthase is an important enzyme. This is a mitochondrial enzyme. This enzyme is going to use it's going to be responsible for forming our carbamoyl phosphate. This is where we take our nitrogen, our NH3, that we just transported into the mitochondria, and we're going to take the ATP and HCO3-, and we're going to make carbamoyl phosphate. Now, what I want you to know about this is I want you to know the building blocks. So HCO3-, ATP, NH3. Those are your 3 big building blocks. That's going to produce a carbamate, and that carbamate can get phosphorylated to make our carbamoyl phosphate. This enzyme, CPS1, carbamoyl phosphate synthase. This is something that is we've talked about another enzyme, CPT1 and 2. That's carnitine palmitoyl transferase 1, carnitine palmitoyl transferase 2. Those enzymes are involved in the production of, or the transport of fatty acid ACPs into the mitochondria. Carbamoyl phosphate synthase 1 is a mitochondrial enzyme, and its purpose is to make carbamoyl phosphate. There is also a CPS2, which we will talk about a little bit later when we talk about nucleic acid metabolism. So what I think is, very important, and we're going to do this over a couple of days, is we're going to break down different parts of the urea cycle. But I recommend looking at this multiple times and kind of processing the information into different chunks for yourself. All right, well, I hope this was helpful, and I hope you have a good one.